It's my great pleasure to introduce the next speaker, that's Alvaro Sanchez. Alvaro, can you start sharing your screen? In the meantime, I'll present a very short introduction. Alvaro is a tenured group leader at the National Center of Biotechnology in Madrid. In fact, he's only recently moved to Spain. Before that, he was an associate professor of ecology and evolutionary biology at Microbial Sciences Institute at Yale. And the Sanchez lab studies the evolution of microbial communities, for which the lab cultivates microbial communities in many defined environments in high throughput ways and develops computational tools to model the interactions and the environment. And today, Alvaro will be talking about the team's work in engineering microbial consortia by exploring functional landscapes. Over to you, Alvaro.
Well, thank you very much. Yes, as I said, I recently moved to Madrid, but for the past 6 years, my lab was at Yale, and what we've been doing and continue to do mostly is trying to figure out how we can, understand how microbial communities form, how they evolve together, how they function, with an eye towards engineering them. And today, I want to tell you about the recent work we've done, trying to understand how microbial community engineering could be done by exploring functional landscapes and how these landscapes can be understood from a set of simple rules. Just a brief introduction for why we care about microbial communities. The main reason is microorganisms are everywhere. They've colonized every habitat on Earth. You find them in essentially every body of water, in the oceans, the rivers and ponds. You find them in very, very large numbers in the soils. And that's only the free-living microorganisms. The bodies of all animals and plants, including our own, are also teeming with microorganisms that co-inhabit our bodies and have very important roles in our health. And beyond the free-living and host-associated microorganisms, one finds microorganisms as well in pretty much every structure humans have built, from buildings to machines, and also in the food we eat. And in all of these habitats, microorganisms play critical roles. They are responsible for a large chunk of the biogeochemical cycles that keep the biosphere healthy and running. And when associated with animals and plants, they are, as we all know, causative agents for health and disease. So, and this is just the microorganisms that live in the wild, but humans have also been harnessing the metabolic capabilities of microorganisms for millennia, primarily for food production, which we have been doing from at least since the dawn of the Neolithic era. But more recently, we've been harnessing the powers of microorganisms for other applications, from cleaning up wastewater to producing biofuels from plant crops. And traditionally, we have been studying microorganisms by isolating them from their habitats and growing them in petri dishes like these ones, or in flasks. And we have been doing that so by studying clonal or semi-clonal populations of microorganisms, one species at a time. And this approach has been incredibly fruitful and has led to pretty much everything we know about microbiology, or most of what we know about microbiology. But as one important feature of microorganisms is that you will very rarely find them forming monocultures of clonal or semi-clonal populations, right? They live in complex ecological communities containing large numbers of species that interact with each other in really fascinating manners. And when I described before all of the services and important functions microorganisms provide, not only to humans but to the entire planet, microorganisms are tiny, right? So those are the collective function of large populations of these cells, and which are typically diverse ecologically, right? So the composition of these communities is quite important for all of the services and functions microorganisms provide. So this is leading to a growing interest in trying to figure out how one can manipulate the composition of these microcommunities towards with particular goals in hand to optimize the services and functions they provide for their use in biotechnology primarily. So, here's the question, right? Like, okay, we want to optimize the composition of a microbial consortium. We want to design a microbial community to provide a particular, in an optimal manner, a particular function. So one could start from a list of candidate species, like say I give you a petri dish with about 100 colonies, each of them representing a single genotype growing in a semi-clonal population. And I ask you, okay, here's 100 strains. Which ones should I inoculate together in a bioreactor to produce the most biofuel from say sugarcane, right, or some other crop? So that question is very hard to solve, right? From out of 100 strains, what could form at least about 10 to 30 combinations, right? And that's only including presence absence, let alone the initial abundances of the different microorganisms, right? So the problem is really vast, and it's difficult or near impossible to solve this computationally and let alone experimentally, right? We cannot simply not form that many communities. So this function, right, of given a list of candidate species, which one should we choose to form a consortium to optimize a particular community service, is one that appears across all areas of applied ecology. Then, and just an example here, going beyond microorganisms and instead focusing on bacteriophages, which are these viruses that infect bacteria. There's a strong interest in trying to figure out which combinations of these bacteriophages could be used to fight and to eliminate bacteria, right, that are resistant to antibiotics or other treatments. But it's still very unclear which particular, if you have a list of candidates, which phages should be combined most efficiently to do this. And beyond microorganisms, if you move to larger systems, for instance, plants and agriculture, there's also a lot of interest in trying to figure out which particular set of crops should be mixed together in intercropping systems to optimize yield or to suppress pathogens, improving soil quality in the case of cover crops, and so on and so forth, right? This is a question of, given a list of candidate species, which one should we use to form a community that optimizes a particular service? It is one that appears throughout many different areas in applied ecology. Now, this is I'm trying to emphasize this is an important question we need to figure out, but to do so, to be able to provide an answer, right? It's going to be important that we are able to quantitatively predict the function of a community from its composition. And when thinking about how people have gone about solving problems like this, and we thought of a similar area that has dealt with this degree of combinatorial complexity, which is genetics. And it's often the case, for instance, in the field of protein engineering, that folks are interested in trying to figure out which particular mutations they should introduce to an enzyme to improve its catalytic rate, for instance, or to improve its thermal stability, and so on and so forth. And just as is the case for the question I was asking in ecosystems, the number of possibilities is also astronomically large. So there are, however, tools that geneticists have developed to do so, to infer the relationship between genotypes and phenotypes, right? Between the structure and the function of their systems. And these tools exploit the fact that there exist regularities in that map, right? In that those regularities mean that local information about the structure of those landscapes can predict more global features and help you find or get close to be able to predict the phenotypes of genotypes that you have not measured yet. And there's been a number of approaches to try to infer the structure of fitness landscapes from an incomplete and even very much incomplete subset of measurements, and some have been rather successful. So what we think is that if it were possible to map this problem and find an ecological analog to a fitness landscape, then it might be possible to import all of these methodologies and ideas from the field of quantitative genetics to ecology. And if that were possible, it might be then feasible to predict the community function from composition and to infer the structure of those landscapes or ecological structure function landscapes from a subset of a very, very much incomplete subset of measurements, right? And with that, predict what the shape of that would be and help us locate functionally optimal consortia or communities that could be thought of as being peaks in these landscapes, as well as exploring these landscapes through directed evolution and so on and so forth. And if we knew more about what the relationship between community composition and function is, we could maybe navigate them using various tools that we could then borrow from the field of genetics. Now, to illustrate the idea, I think it's helpful first to make sure everyone follows me on or is on board on how people do this in genetics and how the problem is approached there. So typically what we would have here is a background genotype, right, consisting of maybe multiple loci, and those loci could exist in multiple different states, right, depending on what different mutations that you could place there. And that gives you the collection of those gives you a genotype, and each genotype is associated to a fitness or a phenotype. And now we can introduce a mutation in one of those particular loci, and that mutation is going to have some effect. It's going to change the fitness or the phenotype of the genotype upon introducing it. And the difference between the fitness effect of the genotype plus the mutation minus the fitness minus the genotype the background, right, that doesn't have the mutation, that is the fitness effect of the mutation. It's the change in fitness upon introducing a mutation into a genotype. And the fitness effect of a mutation, at least considering that the environment is fixed, can be expanded into an additive component, and the sum of an additive component, and as well as the sum of every possible pairwise interactions and third interaction between every possible mutation or that loci and every other loci in the genome. So the additive component, that would be, you could think of this as being the on average, what is the effect of that mutation when you average over all backgrounds. And when you think of these pairwise effects and higher interactions, all of that captures the variation that exists around that average, right? So in the case of a simple fitness landscape, what we would have is that you could have when you have, say, a set of mutations of size 2, we have mutation A and mutation B. The interaction, basically the additive component of mutation A would be this increase, right? And that additive component is going to be the same regardless of what background you put it in, right? If you put it in the null background, you're going to have this increase in fitness, and if you put it on a background that contains the mutation B as well, it would be this, right? This here, right? And the two will be the same. Now, any deviation you see from that, that's the pairwise epistasis, which is the interactions between mutation A and B, which cause a deviation from the null additive expectation. Now, how will this map to the case of communities? Well, in the case of a community, we could think of a community state containing 3 different species. That background community will have a particular function of interest, which could be, for instance, in the case of a plant community, could be the ratio of nitrogen to carbon in the soil, the productivity, the above ground biomass, the harvest yield, or whatever it is that you're interested in that this community is doing. And if we include also in this, and when we see this community, we also include species I, right, into the mix, then that would change, or that could presumably change the function of interest of the community by some amount. And that amount, that difference between the function of a consortia that includes species I and the function of the equivalent community that does not, we can call that the functional effect. By analogy to the fitness effect of a mutation, this would be the functional effect of a species in a community, which is how the function of the community changes when that species is present. And we could then take the same approach and model a fitness effect in the same manner. We could partition the fitness effect, the functional effect of the community of a species, right, as an additive component, which would reflect on average the functional effect of the species across many different ecological backgrounds. And then the deviations from that could be modeled in terms of the sum of every possible pairwise and higher order interaction. Right? And this is work that we have done in the past, and we have published a few papers on this approach. So what this is nice is that it helps us map the problem of understanding of modeling the functional effect of a species in a consortium to the fitness effect of a mutation on a fitness landscape. And just to give you an idea of how we approach this problem and in the context of ecology, I wanted to very briefly give you a brief overview of an experiment we did a few years ago, where in this case, we took 6 different soil bacteria, all of which belong to the phylum Bacillota, and they all secrete an enzyme, which is an amylase to the environment, and that enzyme breaks down starch. And what you see here in this, these are the colonies, right, for each of the species, and this halo you see around them, that's the area, that's basically the starch that has been degraded by the enzymes that the bacteria are secreting outside, right? So in purple, that's the starch, and in what you see here, the halo is the lack of starch. Right? So as you can see, all 6 of these species can secrete amylases to the outside world. And all of them can grow on their own fairly well under the conditions of our experiment, except for this one, Penebacillus polymixa, which is a poor grower because it is a biotin auxotroph. It cannot synthesize all the biotin it needs to grow, so that limits its growth in other growth conditions. Now, every other bacterium here can also support the growth of P. polymixa by cross-feeding. Either we are not sure if the cross-feed is because they provide the biotin, or more likely they are supplying a precursor in the biosynthesis of biotin by P. polymixa. But be as it may, what we see is that this bacteria, P. polymixa, which is a very strong secretor of amylase, but a poor grower, grows very little on its own, but much more so when it grows in co-culture with every other species in this consortium, because they all can facilitate its growth and rescue its auxotrophy. Okay, so now with that introduction, we could quantify the amount of the amylolytic activity of each of these microorganisms, as well as any community you can form from them. This is straightforward to do. You could grow either each of these species separately or combinations thereof in media that contain starch. And after 48 hours, you could just simply spin down the cells, filter the liquid, right, the spent medium, and then put different aliquots of that spent medium into test tubes that contain some amount of starch, and then you can see how starch gets degraded over time. And here on the y-axis, I'm plotting the fraction of starch that gets degraded as a function of time. And what we can do then is we can fit the kinetics of starch, enzymatic starch degradation by the enzymes released by every species separately, or consortia formed with them. And we can then fit it to a 2-step model, which is essentially Michaelis-Menten kinetics. And with this, a single parameter, which is the velocity of the reaction. And that single fitting parameter is the amylolytic velocity of the starch degradation, and it's the function that we're going to be focusing on in this experiment, right? This is the function of the species and the consortia, the velocity of the amylolytic reaction of the enzymes they secrete. It's the amylolytic activity. Now, for all of these species, except for P. polymixa, everyone else, the other 5, we observed that if we quantify the amylolytic activity of one of the species, say here B. megatherium, the other B. thuringiensis, or the combination of both, which is here, and if you grow together in a consortium both megatherium and thuringiensis, what you see is that the amylolytic activity of the consortium is very, very close and almost indistinguishable from what you would expect from an additive model. What would that mean? Is that if the 2 bugs were grown separately, and then you simply took the supernatants and mixed them, right? So there's no interactions between them. This is what you would have observed, right? Is here in gray. And now what is maybe a bit surprising is that when you co-culture these 2 microorganisms the amyloid activity of the pair is basically exactly the same as the sum of the contributions from each in monoculture. Apparently there's no interactions, right? An additive model works very well. And it's possible to show through simple biophysics that precisely this additive model is what you should see if there's no interactions at all between the two organisms or the enzymes they release to the environment, right? So that's very nice. This is what these kinetic curves look like. In the red, those are the data points. The black line is the fit, and the shaded area is the 95% confidence interval of what you would expect if the two species were additive in the function, right? So this is just other words to say this is very boring. Everything is additive, and there's no interactions. And that expands to every consortia you can make, right? So a simple additive model. So basically, if you just know what is the function of every component, right? Then you could predict extremely well what's going to be the function of any consortia you make with them, right? So because you have an additive model that tells you exactly what it should be. So basically the sum of the contributions from each species. Every species acts as if the others weren't there with them from the perspective of amylase production. And then we can very, very well predict what's going to be the function of all the consortia we can form from them, right? Because it's a very smooth landscape, if you want to see it that way. However, when people in Mixa is present, it's a part of the consortia, what we see is that the amylolytic rate of the consortia deviate significantly from the additive prediction, right? So this is what you would expect. And whenever people in Mixa is part of the consortium, then the function of the consortia is far, far larger than the additive model would have expected. And what we tried to do, when we established that is because of cross-feeding and facilitation towards polymixa. But one thing we found was that if you just try to predict if you try to now, because the additive model fails, if you try to incorporate the pairwise interactions as well, and you compare the expected function when the model includes also pairwise interactions with the measure, you also see that there's also very large deviations. Those deviations still persist if you include third-order interactions and fourth-order interactions. It's just basically not possible to predict the function of a consortia through this model, right? It doesn't work. So what we then tried is, in the past couple of years, we've been playing with another way to look at this problem, which is a perhaps more modern view of genetic interactions, which is gives a different way to partition them. And there's been, for the past couple of decades, but more intense in the past few years, some very exciting work on a phenomenon called global epistasis. And global epistasis reflects the fact that the fitness effect of a mutation can be well predicted by the fitness of the background, right? An example is diminishing returns. It's a phenomenon called as diminishing returns epistasis. When you place a mutation in fitter and fitter backgrounds, its fitness effects become smaller and smaller, right? So a beneficial mutation will have a smaller effect when it's placed on an already very fit background, and a much larger fitness effect when it's placed on a low fitness background, right? That's diminishing returns epistasis means. And it reflects interactions between the mutation and every other, every other mutation that you are putting into that, every other loci and every other mutation you can introduce into that genotype. Quantitatively, the global epistasis has been defined by basically doing a regression, quantifying how much of the variance in fitness effects can be explained by the fitness of the background through a simple linear regression. And so the regression line is essentially the fitness effect of a mutation I in genotype G is the component of that is expressed, that is captured by this regression or that linear fit. That's called the global epistasis, whereas the residuals that the fit doesn't explain, that's called the idiosyncratic epistasis of mutation I on background G, right? Each of these points corresponds to the same one mutation placed in, I think it's 91 different genetic backgrounds. Here they're plotting the fitness effect of that same mutation on each of the backgrounds as a function of the fitness of the background, right? I mean, you can see it's this very, very nice linear regression between both. And there's been really fascinating work done by Michael Desai's lab in the past couple of years has actually shown that these slopes, that these regression slopes that people are observing, are actually can be written in terms of the pairwise interactions and the fitness effects of the mutation, right? So basically, the slope of mutation I, then this in the scroll epistasis slope, can be written as a sum over the average pairwise epistasis between mutation I and every other mutation on every other loci, if you want to call it that way, on the genome, divided by the average fitness effect of every other mutation, right? So it is, in other words, what's very nice about this is that the number of potential genetic interactions that you can have, including pairwise and higher order interactions, are very, very large, right? And they explode as the system increases in size. But what they find is that all of these different interaction, genetic interactions you could have, actually converge onto this one parameter, this slope, and these all of these. Then you don't need to measure them, right? You could simply look at the shape of this slope, and that can explain a lot of the variation that you see without having to measure every interaction parameter, which, as I said, is there's as many interaction parameters as you can possible state in your fitness landscape. And just to give you an example of how we could do this is another experiment, perhaps easier to understand. This is a complete landscape formed by 5 different beneficial mutations that fixed in the long-term evolution experiment. There's a very 30-year-old experiment that was started by the lab of Rich Lenski I think it's in 87. And they observed there were 5 mutations that fixed, right, in this adaptation of E. coli. And what they did then is that they reconstructed every potential combination of those 5 mutations, the 31 possible combinations of those 5 mutations, right? Everyone independently, every pair, every trio, et cetera, right? In such a way that every mutation could be placed in 16 different backgrounds, right? So what they're going to do here then is that they plotted the fitness effect of the mutation, each mutation, in this case, this mutation, in each of the backgrounds as a function of the fitness of the background, right? And what they found was that, consistent with what I just showed you before from the Scilab, there are simple regression lines that can capture a lot of the variation in fitness that they were observing, so that the fitness effect of mutation could then be in part predictable from the fitness of the background. Interestingly, they also see that different mutations have different slopes, right? Some, for instance, the pyruvate kinase has a positive regression slope. Other mutations have a negative slope, and they are consistent with diminishing returns. This would be consistent with accelerated returns, where the mutation has a stronger beneficial effect when placed in very fit backgrounds, and a smaller fitness effect when placed in low fitness backgrounds, right? As I was saying, the phenomenon known as global epistasis, which is this fact, right, that the fitness effect can be explained by the background fitness, at least in part, has been very ubiquitous, observed in yeast fitness landscapes, more work from the Scilab, as well as in other bacteria. This is work done by Chris Marks and Daniel Seger's groups 10 years ago, right? And again, they have again observed that the particular slopes of these regression lines can be different for different mutations. So what we wondered was, okay, this is very nice for genetics, but would it be possible that there's an analog idea to this global epistasis observation in ecological communities that could help us predict the fitness, the functional effect of a species in a consortium from knowledge of the function of the background where that species is being added? And so here's the idea, right? We took data that had already been published. This is from a paper from 12 years ago, where they constructed a semi-complete set. I think in fact this was complete, a complete set of every possible background community formed by 6 different bacterial species. And so basically every monoculture, every pair, every trio, and so on and so forth, as well as all 6 together. And they did the equivalent experiment where we placed they looked at the effect of adding the same species to each of the combinatorial consortia that they could potentially assemble. And here, what we could plot from this data is we could plot the functional effect, the change in function, in the function of the consortia upon adding Flowbacterium, as a function of the background community. And what we see is that very similar to what people have observed in genetics, there are global epistasis-like patterns in communities as well, right? Where the effect of including a community member in the consortium can be in part predicted by the function of the background, and that those the particular regressions that predict that are different for each of the species. Each species has its own characteristic slope and intercept. So I show you this one example, but you may wonder how general this is. So we went on and started analyzing other data sets from combinatorial ecological communities whose function had been assessed. In this case, we found this paper where they assembled they seeded plant communities from various combinations of 8 different plants. That is not complete, but they did a bunch of different combinations, pairs and trios and so on. And this is the outcome, right? This is the function that they're measuring is the above ground plant biomass of the entire community. And here on the y-axis, they're plotting we are plotting the functional effect of adding each species to the community as a function of the function of the background community to which you add each species. As you can see, then there's a strong, these linear models explain in this case a very large fraction of the variance, and they work very, very well, right? This is the equivalent of global epistasis, but in an ecological sense. This is another experiment. This is the organisms where communities formed by phytoplankton. The function of the community was the total amount of biomass produced by the community. And these are these global epistasis-like patterns of several of the potential members of this consortia. As you can see, some have a type of epistasis known as increasing costs, right? Like whereby their effect is negative, and it's more negative the more the higher the function of the background is. Others, like Synechococcus, have diminishing returns. It has a positive effect, but its effect is smaller and smaller the larger the function of the background community is. There's one example. This is the full example of the bacterial silos oxidation I told you before. Some species actually are very flat. Others have diminishing returns. Others cross the negative side and become enter a region of increasing costs. And this is for the starch, the amylolytic bacteria I told you before. Remember the consortia of 6 soil bacteria that secrete enzymes, amylase. And these are the patterns that we observe for each of the individual bacteria when we place them in different. We reanalyze the data essentially that I showed you before. So none of these are new experiments. We're just analyzing data from the literature, including our own. And as you can see, some species have increasing returns, like Polymyxa and Bisattalis. Others have increasing costs, like Biserius, for instance. It does contribute to. It's an interesting case, right? Because Biserius produces amylase. It cross-feeds Polymyxa, right? So it has all the traits that naively you would think could be conducive to have a positive effect on function. Yet it has a negative effect, and its effect is more and more negative the more the higher the function of the community you're adding it to, right? And Polymyxa has two different slopes, and if anyone is interested, I can tell you why. It's actually kind of cool. More data we analyzed. This is data from a paper where, again, combinatorial, not complete by any means because there's a very large space, but they made a bunch of different constructs. Very nice paper, by the way, where they looked at consortia for producing butyrate, where you can similarly determine the global epistatic patterns for the various members of this consortia. Again, each one have their own unique patterns. And this is the final experiment I want to show you. This is, again, a reanalysis data that we had from our lab, actually. In here, what we looked at was communities that were not combinatorial consortia grown for a single batch, as were the case for essentially every example I've told you before. These are what's called enrichment communities. So these are essentially natural communities that are grown under serial passaging in the lab. So these are fairly diverse communities. Communities stabilize after 12 passages, so where you grow you basically throw whatever is on the soil. We're talking about thousands of species. you let them grow in a habitat that contains, in this case, citrus as the only carbon source. Most species go extinct, but you still have a fair number of a few dozen species in some cases coexisting in a stable manner right after you passage them multiple times. And after the community is stabilized, what we can do is we can have a library of 8 different bacteria, which are all these 8 isolates that we placed in, and we can place each of those isolates in each of the communities. And after we add them, we allow the consortia to again stabilize, right, by passaging them again. And even in that case, right, which are in a very different ecological regime, and when we measure the function, in this case, is the amount of pyrethrin produced. Pyrethrin is an iron scavenging molecule that microorganisms secrete to the environment. In all cases, you again see that these patterns are there. They explain a substantial fraction of the variation in the functional effect of each species. So it is possible to predict the effect of a species on a community function, even when the communities are not combinatorial or necessarily small, right? So, given that, I hope I convinced you this is actually a very general phenomenon in ecology. In fact, the question is whether it could simply be a triviality, right? And there's reasons to suspect that it might be, right? Or it's not unreasonable to think so. And the reason is that, in fact, if we had a purely random landscape, we should expect to see diminishing returns, right? This fact is clear, right? Because if you have a landscape, right, and you are on the peak on the global peak of the landscape, and any perturbation, adding any new species can only bring you down, right? Because it's already at the peak, right? It is the same thing in genetics, right? If you are, on the other hand, at the lowest point of your landscape, any mutation you make, every perturbation you make in the case of ecosystems, there's nowhere to go but up, right? And the same will be true in a statistical sense wherever you are, right? So the fact that you should see that, the fact that one sees diminishing returns or a negative slope between the fitness effect and mutation fitness in the background is in fact, or could be, simply a statistical triviality. So, but that one, I think, interesting observation is that, yes, we have seen diminishing returns or increasing costs many times, but often we've seen patterns that are not consistent with that, right? So that suggests that it's just not a something trivial. But to make sure, what we did is that we did an experiment. This time, this is the first experiment we actually did that I'm going to tell you about. Juan, a fantastic postdoc in my lab, what he did was he assembled various consortia with soil bacteria, again using pyrethrin production as the function. He measured the function for how much pyrethrin was produced by each of these consortia, and he then randomized the association with the community composition and function, right, creating de facto random landscapes. When you do that, right, and indeed every species has a negative slope, a negative fitness of functional effect versus function of the background, right? There's the change in pyrethrin composition upon adding these species, the Pseudomonas, to its landscape. Again, this is after we randomize the landscape. This is not the actual observation here. It's just when you randomize the association. That one of the things we see is that all of the species would have the same negative slope of -1, the same R-square of 0.5, and essentially everybody would have the same pretty much the same global epistatic patterns if you randomize, right? So if you randomize many times, what you see, this is 1,000 randomizations. You should see all of them are within this cloud here, and this is in gray. So all these colors represent the actual intercept and slope for the regressions for the species in our empirical data without randomization. Just to show you that the actual regressions that we see, the patterns we are seeing, are not a trivial consequence of regression to the mean, right? They contain signal about the particular interactions they have. Okay, so we are then moving on to asking whether these global epistatic patterns will also emerge, like it is the case in genetics, from pairwise and higher order interactions averaged out in a particular way. And what we did is that we applied this equation, which is the same equation I showed you before. It was derived by Desai and Reddy and Desai for the case of fitness landscapes in genetics, and tried to see to what degree we could predict the fitness effects, the global epistatic patterns that we observed in our ecological experiments. And what we find is that, in fact, this equation does an excellent job, right? It really nails it. And what it suggests is that when you take every possible pairwise and higher order functional interaction that exists between the species in our consortia, which again, there are many of those terms, and you don't need to measure them all though, right? Because their effect condenses, funnels through this emergent parameter, right? B, this is the slope of this regression that explains a lot of variation on its own, right? So I told you about this, but I wanted to start closing this talk by telling you a bit why this is useful. First of all, I think it's useful because it allows you to predict what's going to be the effect of adding a species to a community, right? If from just by placing these species in a bunch of different ecological contexts and determining what is the slope that describes it, and then I will be able to predict what's going to be the effect of adding it to other out of sample communities. So that's already very useful, right? But another perhaps more exciting phenomenon is a consequence of this is that if we happen to know the functional effect regressions, right, is the regression lines for each, say, a number of species, then it should be possible to predict the function of any combination of those, right? Any consortia we could make from those species, right? The procedure is simple, right? I could simply take, and with this, after having characterized this training set, right, of consortia, I could say, well, if I wanted to determine the function of the trio of these three species, and without them, that trio being part of this set at all, this is a completely new experiment. What I can do is I could say, well, if I add this one species to an empty vessel where there's not, the function of the background is 0 because there's no background community, I can get what that is going to be, and that's going to be the argument, the function of the monoculture, going to be the argument for when we add the next, virtually add the next species to that consortium, and then I can concatenate that as many times as I want, right? We have to be a little bit careful with the residuals. Again, there's some technicalities here on how to do it properly, but it's possible to do, right? We can simply stitch together these functional effect equations and use that to predict what's going to be the function of new consortia we haven't assembled yet, that are not part of our training set. To test the validity of this, Juan actually did an experiment. He went back to this set of 8 different bacterial strains. 5 of them, the Pseudomonas, produce pyverdins. The 3 Enterobacteriaceae here, they don't, right? They don't produce pyverdin. And he assembled, I think it was 60 different consortia. And then to each of those consortia, he added, so the space of 8 possible consortia is 255, right? 56 if you include them all. But he assembled just a subset of them, like I think it was 60 of them. And then to each of those, he added either one of these species or that species or that species or that species, right? So that he determined what's going to be these what we call the functional effect equations, these regressions for each of the species when we add to every consortium, right? Armed with those, what he did then is he then concatenated them in the way I described before and predicted what would be the out of sample, what would be the function of a number of 60 additional consortia that where he had not assembled yet, right? This is really out of sample in the sense that he would have to go back to the lab and do a new experiment to test whether he could then use this method to predict what's going to be the function of those consortia. Again, it's very important under the same environmental conditions, right, as the training set. So if things were working well, right, if this method were working well, if you plot the predicted amount of pyverdin in the environment in micromolars against the observed amount of pyverdin produced by our out-of-sample communities, the newly assembled ones, they should cluster around this identity line, right? Where the prediction should match very closely what's observed. And that is in fact the case, right? This is a new experiment with consortia that we had not assembled before, but we're able to predict what the function is going to be very, very well from this very simple idea. And then, so this is an experiment we did in triplicate, multiple biological replicates and so on. It's very reproducible. But then we could see if, I mean, with the data that has already been published, we cannot do that, right? Because data was published and a lot of the times it didn't do replicates and things like that. But we could just simply ask to what degree this approach would work if we take some subset of the data as our training set and the rest of the data as our test set, right? And we did, right? This is for several examples we took from the literature. This is the bacterial cells oxidation data. This is actually the predicted versus observed function for our amylolytic communities, which we cannot predict by simply just summing up all the terms of Bergman's epistatic terms. But through this approach, we can predict very well. This is for the nutrient secretion data from the Ventrally lab. This is for the plant consortia. So again, I think it's very nice. Again, this is more the training set is small here, right? Because they didn't have a lot of communities they tried, so it's harder to get good statistics. But still, I think the outcome is very promising. And this is for the phytoplankton data. And all these are experiments from the literature, which we simply reanalyzed. Some good news is that because of these linear patterns can be very successful in helping us to predict function, even when they're so we don't need a lot of background communities to make this work. Particularly if we're interested in finding what are which communities are going to be closer to the optima, even if not the absolute global maxima. But if we're happy with being very close to it, then this method works very well. You can get very close to if you calculate the ratio between the function of predicted optimal community and the actual optimal community, you can get very close, even if you subsample substantially and just have, in this case, as few as 3 communities to train what's going to be the functional effect equation, right? I think this is interesting for many reasons. I particularly like this the sentence that Juan wrote, that this approach brings the task of predicting biological function at the genetic, organismal, and superorganismal or community level under the same quantitative formalism. I think that's very exciting, the fact that this is starting to look like it's working. But beyond that, I think I wanted to reemphasize that there already are a lot of tools that geneticists have built to exploit the existence of fitness landscapes and to navigate them and find optima in them, right? So if we could borrow those techniques to the ecological setting, now that it's clear that this the concept of a community functional landscape could potentially work again under pretty much the same circumstances when genetic fitness landscapes work, right? So it's under the same environment, right? So if you change the environment, a genetic fitness landscape maps genotype to phenotype in a given environment, right? A community function landscape also maps community composition to function in a given environment, right? There's a lot of caveats. We have written a piece that hopefully will be accepted soon, where we have overviewed under what conditions the community function is encoded in the composition. It's not always the case that it is, but I'm happy to send the link to anyone if anyone's interested in reading more about this. But the broader point is that if such a map exists, and there are already tools we can borrow from other fields to help us navigate these landscapes. And as I was saying before, there really are many tools that are being developed and which could potentially be a game changer, I think, in the design of microbial consortia. We have been also very interested in other ways to explore these landscapes, not necessarily by mapping them, but from a more top-down approach, by applying directed evolution or artificial selection to navigate them. And basically through multiple rounds of selection, stabilization, and randomization, we have demonstrated through simulations as well as now experimentally that one can navigate these landscapes even when you don't necessarily know what structure they have. It's still possible to navigate them. So again, bringing ideas from evolutionary biology and evolutionary engineering primarily to the field to the purpose of trying to engineer microbial consortia could be very, very fruitful. And that's it. This is work that I've done with the fantastic folks in my lab, to whom I will be forever grateful, as well as a group of wonderful collaborators that have really enriched our experience. And that's all I wanted to say. So if anyone has any questions, I'll be more than happy to to address anything.
Wow, thank you. Thanks a lot. This is an amazing talk. I see applause from Xiaojun. Are there questions? In Augusta, Matthias?
Some questions?
Moment. Okay, perhaps I'll hand over to you, Manish, for the moment. And if there are questions, I tell you.
Yeah, I don't see any question in the Zoom, but I mean, so I think you hinted at it towards the end when you said, I mean, this analysis is relevant for when your resource set or the environment in this case stays the same. But what is the effect of initial inoculum? Like when you do it experimentally, are you adding, similar ratios of these different species, and how does that affect the final outcomes?
Right, that's a really good question. When you do single batch communities, what we have done is we have always kept the same number of cells from each of the species, right? And that practice has been also the case, I believe, when in the experiment we've analyzed. And I do not know to what extent things will be different if the number of species had changed. We haven't done it, and I'm not aware of anyone having it done. That's certainly one of the things we need to explore. But one of the things that I think is quite remarkable is that when we have in this experiment here, here, right? We had the communities were in equilibrium, and we added the bacteria, and then we let them stabilize. In some of these communities, the bacteria we added, the knock-in isolate, they didn't make it, right? So community stabilized, and the isolate went extinct. So I am still struggling to understand why this works, despite the fact that the species you put in isn't even there at the end of the experiment. But at least this suggests that it might not be that as critical as you might think. Again, this is one experiment. We definitely are going to repeat it, and this is data analysis, right? So this experiment was not done for the purpose of that. It's experiment published in the context of another paper. We simply reanalyzed it. But I think the fact that this works as well as it does suggests that perhaps presence absence might be enough, even in that context. When I've been asked, when people have asked me about how is it possible, right, that we could predict the function of the consortia without knowing what the final population size of the species is, in some sense, in the theory of fitness landscapes, right, when you say introduce a new gene into the cell, if you had done the same thing for genes, right, and we know that this global elasticity approach works, right, but we don't need to know how much it was expressed, right? So you could put a gene, and the gene might not be expressed too much, or in some context more, in some context less, but still, right, these global epistatic patterns are seen. So I wonder if it's related to that, but I do not have a good explanation for why, right, the presence absence works as well as it does. But I can tell you we're actively working on it, and hopefully I'll be able to give you an answer sometime soon.
But I wonder, I mean, since you bring this parallel with the, genetic networks, in genetic networks, you're right that the expression may change, but sort of the dosage of the gene puts bounds on the range within which expression can change, right? And I feel like, I mean, again, this is intuitive, and I cannot say that I'm certain of this, but it seems to me like in communities growing, these bounds are much broader, and you could go from one member of the community exploding, whereas,
Yeah, I agree. I mean, I am very surprised by this, and the fact that even when the final species isn't even there. Again, this is one. In an experiment, it wasn't even done for this purpose. Basically, just the analysis. It could have been a fluke. I do not know, right? That I think it's an excellent question. I just don't know what the answer is, right? I just brought this up because I think it's quite interesting that, at least in this case, it seems like that might not be as important, right? But I don't know if it's general, if it was just for this particular case for some reason, and in others it would be. We are in the process of trying to understand it. I agree with you. I mean, I think it's clear that intuitively, at least, you think it should matter what the initial dosage is. But to what extent it really is in the end, I am not sure, right? I still don't know.
I wonder, I mean, perhaps not specifically for what you presented, but I wonder if in general in community analysis, there is this idea of some kind of sensitivity analysis in terms of some species, small changes in their, or some members, small changes in their numbers will affect the species more than others. And in that sense, they become key controllers or keystone species in this.
In a functional sense, right?
Yeah, in the functional sense.
Yeah, I think so. I mean, and I think that the slopes that you see are quite telling, right? I think a species that has a very, very large dynamic range, right? It does tell you that its effect can be very large or very small, right? So it's more likely that it'll be, play that role that you're suggesting. Whereas some species that have a very flattish, this isn't the case here in any of these particular ones, but some species we have found have a more flat, right, effect that might be more additive or more system independent, right? And perhaps that's telling you that their interactions are weaker, right? The size of the slope is proportional to the average pairwise epistasis that has, right? So when a species has very large interactions with others, it'll have, or just, in any distribution you want, right? It's the average, right? So it doesn't have to be with everyone, or maybe just one very large and others are weaker. But in any case, when the average pairwise epistasis is very large, the slope gets larger, right? So I think that tells you something about that. But again, I do not know how the final population size factors in here, right? This is an area that we are hopefully going to be investigated soon. But I don't know, what the outcome is. I still think this is very surprising that this works, right? For precisely that reason, right? That some species you might imagine they're going to have higher sensitivity, right? Whereby their increases or decreases in abundance might shape ecosystem function more than others, right? And the fact this is true even in small communities, right? Where perhaps you might think if community is very large, but maybe every one individual species has, less power over that. But the fact is that we have seen this in every single consortium we've ever looked, right? Although, as you pointed out, the majority of them are single batch, right? So it is possible that if we were to analyze this in stable communities, then there we would see that the particular population size will become more and more important.
Thank you. Other questions? I don't see any questions on Zoom. I was wondering, in fact, as you were answering this question, is there studies on sort of molecular level relationships that explain the, community level behavior in terms of metabolic exchange between species, or the total cap on the resource available in the environment? How does that shape this community composition?
Yes, I mean, it will be, right? Like in the case that I described before, let's see here, this is just one example, but there's there's definitely many others where facilitation is very important, right? So like in this case, the secretion of what we think is probably a precursor for biotin production among the other 5 community members, rescues the growth of Pedicillus polymixa, and when that fact, right, the fact that there is this facilitated interactions, meaning this positive pairwise epistasis between each of these other species and P. polymixa, is actually critical for the fact that when you go and look at now this data here, the slopes are positive, right? So you could show that when there is facilitation, then the slopes are more likely going to be positive slopes, where when there is competition for resources, the slopes are negative, right? And the more competitive a species is, the more negative its effect is going to be. The more facilitation it has, the more positive it will have, right? It will be, right? So I believe it is possible to connect the patterns that we are seeing with molecular level interactions between the species. And in fact, I think it could be very helpful to help us identify, oh, there's something here, right? So if you see a plot like this, oh, actually, there's got to be some facilitation going on, right? Or if you see something like this, it's like, well, there's got to be some competition. What could be the origin for that, right? So I think that this kind of analysis can help us also identify mechanisms of ecological interactions at the molecular level that could be responsible for the patterns that we observe.
And since you're on that slide, why are there two slopes? You
Yes.
already alluded to that.
Yes, it has to do with in this case, it has to do with the fact that all of these these two clouds of points differ from one another in the presence of one other community member, which is B3, right? And it's what happens is that when B3 is present, which is in this consortia over here, right? The amount of facilitation required for polymyxa is smaller, right? So the slope declines, but also the main difference is the intercept, right? And the intercept is the fact that they have different intercept comes down to a bit of a statistical artifact, right? Because when you have thuringiensis present, thuringiensis communities have a higher background function, right, than those that lack it, right? So if you have a bimodal distribution of functions in the background, that will lead to multiple intercepts. So in other words, you will see a single slope when the distribution of background functions is unimodal, right? But if you have a distribution of background functions that is not unimodal, where you have different modes in your distribution, that can lead to different regions in the landscape. So ultimately what this means is that the functional landscape have different regions, right? And then in each of those regions, there's what we call, rather than a single global epistatic pattern, where you have is two different regional epistatic patterns, right, that are predictable within it, right? So so this is something we're working on at the moment, which is how do you find when you should expect to see this or that, like something like this, but it's a single pattern, or something like this when there's two, and yeah, this is work we're doing at the moment to see if we can figure that out.
Great, thank you very much.
You're very
If there are no other questions, then now I don't see any. So I'll thank you again for this very exciting talk.